Assalamu alaikum friends. I hope so that you all will be fine. Friends, in this video I am going to discuss about digestion and absorption of protein. Okay, so let's start our lecture. Friends, what does it mean digestion? Digestion means to break down. The breakdown of complex macromolecule into simplest form. And what does it mean absorption? Absorption means to come in blood. In this video we will see that how the digestion and absorption of protein takes place through our gastrointestinal tract. Okay. So the starting site for the digestion of protein, as we know that protein it is the polymer of L-alpha amino acid. And the starting site for the digestion of protein it is stomach. Must remember the starting site for the digestion of protein it is the stomach. Stomach. Now stomach it is an stomach basically it is an organ. Stomach it is an organ which is made up of tissues. Okay, which is made up of tissues. Now, this stomach, it is made up of epithelial tissue and glandular tissues. And the glandular tissue, it is made up of similar cells that perform same function. And basically, there is a tissue that is glandular tissue. Glandular cells. These glandular cells, they release gastric juice. Okay, gland mean to secrete something, so they secrete gastric juice. So stomach secretes gastric juice, and in gastric juice there is basically HCl present, HCl and pepsinogen. Okay, this is the acid HCl, hydrochloric acid. Now, HCl it is very important. It is responsible for denaturation of protein. Denaturation of protein. It is very important. Denaturation means loss of secondary, tertiary, and quaternary structure of protein. Only primary structure remains. Okay. This is suppose this is the structure of protein, and after the denaturation process only primary structure remains, and these are amino acid, because protein it is made up of amino acid, and amino acid they binds each other with the peptide bond. Only primary structure remains. So HCl it is responsible for the denaturation of the protein. Denaturation means loss of secondary, tertiary and quaternary structure of protein. Only primary structure remains. Now this pepsinogen it is the inactive form of an enzyme. Friends must remember that the एंजाइम की पहचान कैसे होती है कि उसके आखिर में ASE आता है. ये एंजाइम की एक पहचान है, लेकिन ये एक्सेप्शनल केस है ये एंजाइम ही, लेकिन चूंकि इसके आखिर में ASE नहीं है, लेकिन ये एंजाइम ही है, ये एक्सेप्शनल केस है. ओके, नाउ पेप्सिनोजेन it activates this pepsinogen, it activates with the help of HCl into pepsin, and pepsin It is endopeptidase enzyme. Endopeptidase, what does it mean? Endopeptidase enzyme mean it breaks down the internal peptide bond. Okay, so stomach, stomach it is an organ which is made up of various tissues like epithelial tissue, glandular tissue, sorry, epithelial tissue and muscular tissue, sorry, epithelial tissue and muscular tissues. It is made up of epithelial tissue and muscular tissue. Basically, tissue it is made up of similar cell that perform same function. In epithelial tissue it is made up of glandular cells, and this glandular cell secrete gastric juice, and gastric juice there is HCl and pepsinogen. HCl it is responsible for the denaturation of protein, and pepsinogen becomes active with the help of HCl. Now, friends, this renin, renin, or we can say another name is chymosin. This renin it is active only in the infants, and this renin it enzyme acts on milk protein that is casein and converts it into calcium paracasinate form, which is the solid and insoluble form. It takes some time in stomach and then it is acted upon by pepsin enzyme. Okay, so these are the events that occur in the stomach. With the help of HCl, there is denaturation of protein, and with the pepsinogen, pepsinogen becomes active with the help of this acid that is HCl. And renin, this enzyme activates only in the infants, and it acts upon milk protein that is casein and converts it into calcium paracasinate form, which is solid and insoluble form. It takes some time in stomach and then acted upon by stomach. Now coming toward another side, that is intestine. Okay, when time, friends, when time reaches to intestine, when chyme reaches to intestine, and then there is release of certain hormones. First one is cholecystokinin. Second one is secretin, and third one is enterokinase or enteropeptidase enzyme. Okay, these are very important. When chyme reaches to intestine, there is release of certain hormones, cholecystokinin, secretin, enterokinase, or enteropeptidase enzyme. This cholecystokinin it releases from duodenum. Okay, this secretin it releases from duodenum and jejunum. This enterokinase or enteropeptidase enzyme it releases from intestinal mucosal cell. Okay. Now, friends, this secretin, what is function of this secretin hormone? It is very important. It acts upon pancreas to release bicarbonate ion, and this bicarbonate ion it is very important. It neutralizes the acidic pH of stomach. It neutralizes the acidic pH of stomach. For pancreatic enzyme to act, it requires 8 to 8.5 pH. Okay. Now cholecystokinin, it acts on pancreas and in this way releases pancreatic enzyme. Okay. So this is very important. In intestine, there is release of certain hormones. This cholecystokinin, it acts upon pancreas and responsible for release of pancreatic enzyme. We will discuss in next moment. Secretin, it acts upon pancreas and responsible for release of bicarbonate ion. It is very important and it neutralize the acidic pH of the stomach. Now coming toward the pancreas, the third side. Pancreas. Friends, there is a release of certain enzyme from pancreas, and first of all, first enzyme, and these enzymes are present in pancreatic juice. The first enzyme is trypsinogen. It is inactive form and it becomes active with the help of enterokinase enzyme or enteropeptidase enzyme. It becomes enteropeptidase enzyme. It becomes activate. Trypsinogen, it converts into trypsin. Okay. Now another one is chymotrypsinogen. This chymotrypsinogen, it activates into chymotrypsin with the help of trypsin. With the help of trypsin, it becomes active into chymotrypsin. Now proelastase. Proelastase, it becomes activate into elastase with the help of trypsin. And the last one, and that is procarboxypeptidase. It also activates into carboxypeptidase by trypsin. So friends, these are the enzymes that are secreted from pancreas, and these enzymes are present in pancreatic juice. The first one is trypsinogen. Trypsinogen, it becomes activate with the help of this enzyme enterokinase or enteropeptidase enzyme. Another one is chymotrypsinogen. It activates into chymotrypsin with the help of trypsin. Proelastase, it activates into elastase with the help of trypsin, and procarboxypeptidase, it activates into carboxypeptidase. Now coming toward the complete overview of the metabolism of protein into amino acid. Okay. So on first of all, there is action of an enzyme, that is pepsin, and pepsin, it is endopeptidase enzyme. This pepsin, it acts upon N terminal of phenylalanine, tyrosine, tryptophan, and leucine. This pepsin acts upon N terminal of phenylalanine, tyrosine, tryptophan, and leucine and converts protein into peptide and oligopeptide. Okay. Now there is action of another enzymes, and that are and that are, first of all, that is trypsin. This trypsin, it acts upon the C terminal of arginine and lysine. Okay. Also, there is action of chymotrypsin, elastase, elastase, and carboxypeptidase. Okay. This chymotrypsin it acts upon C terminal of phenylalanine, tyrosine, tryptophan, leucine, histidine, aspartate, and glutamate. This elastase it acts upon C terminal of lysine, serine, and alanine. Okay, and this carboxypeptidase, it is exopeptidase enzyme. This trypsin, chymotrypsin, elastase, these are endopeptidase enzyme. This is exopeptidase enzyme. It cleaves the terminal peptide bond. Okay, this acts upon only C terminal and converts these peptide and oligopeptide into smaller peptide, dipeptide, and amino acid. Okay, now again there is action of enzyme, that is dipeptidase enzyme and aminopeptidase enzyme, and ultimately there is formation of amino acid. So in this way, that's how the digestion of protein takes place. This is the digestion of protein. First of all, there is action of this pepsin enzyme. Pepsin acts upon N-terminal of phenylalanine, tyrosine, tryptophan, and leucine and converts protein into peptide and oligopeptide. Okay. Then there is action of these pancreatic enzymes that are trypsin, chymotrypsin, elastase, and carboxypeptidase. This trypsin acts upon C-terminal of arginine and lysine. This chymotrypsin it acts upon C-terminal of phenylalanine, tyrosine, tryptophan, leucine, histidine, aspartate, and glutamate. Elastase it acts upon C-terminal of lysine, serine, and alanine, and this carboxypeptidase it acts upon C-terminal and convert this peptide and oligopeptide into smaller peptide, dipeptide, and amino acid. Then there is action of dipeptidase and aminopeptidase enzyme and converts them into amino acid. Endopeptidase mean it cleaves down internal peptide bond, and exopeptidase mean it cleaves the terminal peptide bond. So this is the digestion process of the protein. Now coming toward the absorption of this amino acid. Okay, coming toward the absorption. Absorption. This amino acid it gets absorbed through the ileum part of small intestine. And there are three mechanisms for the absorption of amino acid. The first one is sodium dependent symport. Okay, so अब इसको पहले देख लेते हैं सोडियम डिपेंडेंट सिंपोर्ट. This is the type of secondary active transport, and here the utilization of energy is indirect. I am repeating. It is a type of secondary active transport, and here the utilization of energy is indirect. So what happens? This is okay. There is a carrier protein. This is the carrier protein. Carrier protein. Now what happens that sodium and amino acid, sodium and amino acid, they enters into this intestinal cell with the help of this carrier protein, sodium and amino acid. As they move along in same direction, so it is called as symport. Okay, sodium and amino acid. Now here there is again sodium potassium ATPase, sodium potassium ATPase. Here the energy is utilized and ATP it converts into ADP and inorganic phosphate, and three molecule of sodium they move out and two molecule of sodium they move into this cell, two molecule of potassium ion. With the help of this sodium potassium ATPase, here ATP converts into ADP and inorganic phosphate. Three molecule of sodium ion move out and two molecule of potassium move in. So this is the simplest absorption of the amino acid with the help of sodium dependent symport. Why symport? Because sodium and amino acid they move in same direction, so that's why with the help of this carrier protein this sodium and amino acid move inside to the cell. Okay, and then with the help of sodium potassium ATPase enzyme, three molecule of sodium move outward and two molecule of potassium move inside the cell. Okay, sodium dependent, it is a type of secondary active transport. Why? Because utilization of energy is indirect, so that's why. Now coming toward the another mechanism, that is sodium independent transporter. There are for acidic amino acid there are various transporter. For basic amino acid there are various transporter. For neutral amino acid there are various transporter. For amino acid and glycine there are various transporter, and for beta alanine there are various transporter. Okay. Now coming to the third one mechanism which is very important, that is Meister's cycle. Okay, Meister's cycle. This is very important, that is Meister's cycle. The another name of Meister's cycle is gamma glutamyl cycle. In this cycle, there is a transport of glutamate and cysteine, and three ATPs are utilized. Okay, now coming toward the Meister cycle. Here is this is a cell membrane. This is the extracellular fluid, and this is intracellular fluid. And here is the gamma glutamyl transferase enzyme. With the help of this enzyme, this amino acid move into this cell. This amino acid moves into the cell. Now here, this amino acid, it gets fused with the glutathione, or it gets condensed with the glutathione. The another name of glutathione is gamma glutamyl cysteine glycine. And here the there is a release of cysteine glycine, and it converts into it converts into glutamyl amino acid. I'm repeating. With the help of this enzyme, amino acid moves into the cell and amino acid gets condensed with glutathione. Another name is gamma glutamyl cysteine glycine, and there is release of cysteine glycine and there is formation of glutamyl amino acid. In next reaction, with the help of gamma glutamyl cyclotransferase enzyme, this amino acid, it gets released and it moves into the cell, inside cell, for further metabolism process. With the help of this enzyme, there is a release of amino acid, and amino acid is used for further metabolism process. And there is formation of 5-oxoproline. 5-oxoproline. Okay. Now next reaction, this 5-oxoproline, with the help of 5-oxoprolinase enzyme, and here ATP is utilized. ATP converts into ADP and inorganic phosphate, and there is formation of glutamate. There is formation of glutamate. With help of 5-oxoprolinase enzyme, there is formation of glutamate and ATP converts into ADP and inorganic phosphate. In next reaction, this glutamate, here cysteine gets incorporated into this reaction, and with the help of synthase enzyme, and also here also ATP utilized. ATP, it converts into ADP and inorganic phosphate. Cysteine gets incorporated into this reaction. Cysteine. And there is formation of gamma glutamyl cysteine. Okay, gamma glutamyl cysteine. In next reaction, there is again condensation of glycine. There is condensation of glycine, and here ATP gets also utilized. It converts into ADP and inorganic phosphate. With help of synthase enzyme, there is formation of glutathione, that is gamma glutamyl cysteine glycine. The another name of glutathione is gamma glutamyl cysteine glycine. That is the Meister cycle. 3 ATPs are utilized. First one ATP here, this is the second ATP, and this is the third ATP. Okay. Now I am repeating, with the help of this gamma glutamyl transferase enzyme, amino acid gets transported into the cell. Now amino acid, it gets condensed with the glutathione, another name is gamma glutamyl cysteine glycine, and there is a release of cysteine glycine and formation of glutamyl amino acid. Okay. With the help of this enzyme, gamma glutamyl cyclotransferase enzyme, there is a release of amino acid in this cell. It is used for further metabolism process. This converts into 5-oxoproline. With the help of 5-oxoprolinase enzyme, ATP is also utilized. It converts into the glutamate. With the help of this synthase, that is gamma glutamyl cysteine synthase enzyme, there is the incorporation of cysteine, condensation of cysteine, and with the help of gamma glutamyl cysteine synthase enzyme, there is utilization of ATP and there is formation of gamma glutamyl cysteine. Next reaction, there is condensation of glycine. With the help of this glutathione synthase enzyme, there is again utilization of ATP and there is formation of glutathione. Okay, so this is the Meister cycle. So friends, it is all about the digestion of protein and its absorption. Keep watching and thank you. And if you like the video, then kindly subscribe to my channel. Thank you.